Hello, everyone, and welcome to today's live broadcast, Standardized Precision Medicine, a Quality Management Perspective on NGS-based Individualized Therapies. It is presented by Christoph Endrullat, who is an independent freelancer. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Christoph Endrullat. I will now turn the presentation over to him.
Okay, so welcome everyone. I'm Christoph Endrullat, and I want to say it's a great honor for me to give a presentation here on LabRoots in front of the screens around the globe of a worldwide audience. So I'm happy to share my experience and my expertise with you. I will talk today about precision medicine and from a standardized point of view. So standardization, which plays a pivotal role in a lot of industries nowadays, and the application of standardization and harmonization in precision medicine. So I will give you a quality management perspective on NGS-based individualized therapies. So as I have the honor to be the first presenter today, I want to go in a little discourse on precision medicine. So what does precision medicine means? What are the benefits of it? At first, individualization, right? So individualized treatments for individualized patients, which means tailored treatments. So not a one-size-fits-all treatments. It's more tailored for the unique patient itself. And one great benefit of this precision medicine approach is we have better response rates, we have fewer adverse events, we are able to monitor and surveil resistance mechanisms during a therapy. And also one general aspect, precision medicine offers reduced costs for the healthcare system itself. I will go more into detail during my presentation. But what I want to tell you before, this is very interesting in my opinion also, because I always confuse precision medicine and personalized medicine. I thought that's a common term, so both are used equal, but actually that's not. Precision medicine is not equal to personalized medicine. According to the NIH, personalized medicine implies a unique treatment for each individual. So one single patient, one single treatment. But precision medicine means effective approaches for different patients based on their genetic profile, their environment, and also their lifestyle. So it's rather a detailed or more strong patient stratification than a unique treatment at the moment. And precision medicine is needed. It's time to do this. So and nowadays, where still patients receive a chemotherapy, either they are benefiting or they're not benefiting, they have adverse events. And therefore, it's time to make a change in this field. I put a nice picture here, which I really like. This picture shows an appliance for measuring the feet size. So if you want to buy shoes, you get a personalized approach for buying these shoes. Your feet gets measured, the width, the length, So, and this is quite confusing actually, because for foot size, for buying shoes, we have a more personalized approach than currently in medicine. This is an interesting thought in my opinion. So, and this is what we have. Actually, one size does not fit all. According to the shoes, large shoes will not fit for a smaller person, and the other way around. And that's the same with personalized medicine, also with suites, business suites. Like on this picture, we have this little boy wearing a way too much or too big suite. So, and this has to be transformed into this. So the suite which is not fitting for this little boy has to be tailored that it's fitting. And this is the way we want to transform the medicine today, from the left side to the right side. And how does this will be accomplished? By standardization and harmonization, crucial parts. And I will go into more detail later on. But before, some little bit more discourse on precision medicine. There was a study done by PricewaterhouseCoopers, the strategy and consultants. They asked different precision medicine stakeholders about the expected benefits along the pharma value chain. And those respondents were asked to select up to 5 options, from research, development, market authorization, and post-product launch. And it's interesting what the stakeholders responded to the study. In the research area, most respondents see as a benefit the identification of new drug targets for the tailored treatments. In the development sections, the stakeholders see more convincing data outcomes of clinical trials, also more effective and efficient clinical trials. In the market authorization, the stakeholders see as one of the biggest benefits clarity around the target patient profile, and also an increasing competitive advantage. And finally, in the post-product launch, the stakeholders share the opinion that improved outcomes throughout a continuous therapy are achieved. So which means, for example, monitoring of patient response. But how our precision medicine is performed? We need metrics, we need benchmarks in precision medicine. And there I come to the term of the biomarkers. Biomarkers are rapidly expanding in precision medicine. And the idea behind is we have a patient population. Let's see it on the right picture. We have different patients, and at first a tumor subtyping is performed, that we get a better patient stratification. Then we have different patient populations with different tumor subtypes. And then we can analyze those patients according to subtype-specific prognostic biomarkers. And then we have a much more detailed patient stratification, and we can distinguish between patients who will benefit or who will not benefit according to their biomarker profile. And there's already a remarkable biomarker landscape. We have established biomarkers like the ALK in non-small cell lung cancer. We have the ROS1 also in NSCLC. We have the EGFR in NSCLC, colorectal carcinoma, and also a specific form of the breast cancer. We have the HER2, which is also quite old and famous biomarker in breast cancer. We have the CMET and HGF in NSCLC and different cancer types. We have the KRAS in MCRC, also in lung cancer, and also the BRC1 and BRC2 in breast cancer and ovarian cancer. But this list is not intended to be complete. There are more biomarkers which are established. This should just give an overview. One really strongly accelerating field in the biomarker research are the immuno-oncology biomarkers, the IO biomarkers. As we know, there are a lot of immuno-oncology treatments which have a strong impact on clinical practice and also in patient benefits. And there was a study which was performed by DeCi Bio Consulting, so Decisive Biosciences, they performed a study where they have taken data from clinical trials and analyzed those data. For example, which biomarkers were analyzed within clinical trials. And they have analyzed 1,000 immuno-oncology clinical trials, and they have identified more than 1,200 biomarkers, from which 90% are IO specific. And this is a really nice figure here on the right. Here you can see the number of trials and also which molecular type they are, the biomarkers, either protein or nucleic acids or cells. And the width of the circle there corresponds to the number of trials which are performed. And here in the middle, which catches your eye, you can see PD-L1, typical immuno-oncology biomarker, where a lot of treatments, the upcoming immune checkpoint inhibitors are aiming at. So we have a huge amount of clinical trials, but also you can see HER2, the cytokine profile, estrogen receptor for breast cancer, and also a lot of different, several others. And those are also the next generation, the new biomarkers. They are also called next-gen IO biomarkers, and we can differentiate between the most explored predictive biomarkers currently. Those are tumor infiltrating leukocytes, the PD-1, the TMB. This is the tumor mutational burden, which plays a pivotal role, especially in NGS, which I will come to later on. Also peripheral blood immune cell population and gene expression signatures. Then we have also the most explored response monitoring biomarkers. So once patient receive a precision treatment, the response can be surveilled by diagnostics, and also when mutations occur due to the treatment, this can be detected. And therefore, the most explored response monitoring biomarkers are the peripheral blood immune cell population, gene expressions, and also the cytokine profiles. And now, finally, the fastest evolving biomarkers, so the biomarkers which will play a pivotal role in the future. Those are at first CTC, also the tumor infiltrating leukocytes, PD-1 and the ligand PD-L1 for immuno-oncology treatments, and the genomic signatures again, so tumor mutational burden and MSI, the microsatellite instability. So these are genomic or molecular profiling biomarkers for future advancements in precision medicine. So, but how are these biomarkers detected or determined? There I come to the term of diagnostics. They are the crucial part within a precision medicine approach in order to determine the biomarkers, in order to give a result about presence or absence of a biomarker. Also the quantity plays a role, and also the quality assured analysis regarding the samples, the technology platforms, and the applied procedures. And here on the right, you can see typical diagnostics of biomarkers is performed by pathologists. There's a very nice picture from a breast cancer, which was colored with a dye. And you can see different biomarkers there in these pictures. For example, the estrogen receptor, the progesterone receptor, also EGFR, all crucial biomarkers which are relevant for the diagnosis and also the continuing treatment, in this case for breast cancer. And this now, diagnostics is a different term from companion diagnostics, because companion diagnostics, those are the diagnostics which are officially linked to an officially approved biomarker and also a corresponding treatment. And those companion diagnostics, they should ensure the safe and effective use of a product, so the final treatment, and they can be differentiated between in vitro and imaging technologies, and they are either cleared or approved by the FDA in USA. And the idea behind the companion diagnostics is, you can see on the right picture again, we have a patient population, and for this patient population, biomarker diagnostics takes place. And each different patient populations receives a unique therapy based on their biomarker diagnostics, respectively based on their genetic profile. So, and those patients are most likely to benefit from the treatment without or with really less adverse events. And the old structure or the old strategy of treatments was we have just a patient population, a big one, and they received all one therapy, and then we had patients who would benefit, who had no benefit, or in the worst case, who had adverse events. And the companion diagnostics market has its aim to transform the left side into the right side, and that's the idea behind. And the same with the biomarkers. We have already established companion diagnostic technologies. The most common or used one is the immunohistochemistry, which is used, for example, for estrogen receptors, progesterone receptors, and EGFR in breast cancer, like in the picture I've shown before. Also, the fluorescence in situ hybridization is used, for example, to determine ALK in non-small cell lung cancer. And also the more molecular techniques like quantitative real-time PCR, which is used for determining BCR-ABL1 in chronic myeloid leukemia. And also, and this is what my presentation is aiming for, the NGS, the upcoming technology in companion diagnostics, because this can determine detailed genomic profile, measuring insertions or deletions, copy number alterations, microsatellite instability, tumor mutational burden in different tumor entities nowadays in NSCLC, breast cancer, ovarian cancer, CRC, and also in melanoma. So here you can see NGS plays a very important role in the clinical oncology because NGS is rapidly evolving and it enables a molecular profiling of individual patients. Furthermore, it enables the identification of genomic driver alterations in cancer, which is normally utilized by whole genome sequencing. And it enables multiplex assays. We can sequence different samples in one run, and so the parallelization is given NGS. And also one big advantage is requires just low sample amounts, for example, down to 50 nanogram DNA, which is really important in the clinical samples, especially if we take into consideration the FFPE samples. They have normally a very low amount of DNA and also low quality, but NGS make it feasible to sequence those samples quite straightforward. And so the importance of NGS in clinical oncology is also reflected in the clinical trials model. So we had the past models of clinical trials, normally starting with eligibility assessment and the randomization of the patients. And then we had two cohorts with a control therapy and a tested therapy. And finally, we had the endpoint and data analysis and the comparison. And the NGS, the prospective NGS model in clinical oncology offers new opportunity. At first, after resecting a tumor, an NGS eligibility assessments will take place, and then we can distinguish the therapy A can be given. If we have no response, the original NGS data can be reanalyzed and again assessed for the NGS therapy, NGS-based therapy, and then we can apply the therapy B for these patients. Recurrence takes place after therapy A. NGS offers the analysis of the recurrent site and then subsequently assessment for the next therapy. And then we can give a combination of therapy A plus B. And also the same for occurrence of metastasis. The metastasis can be analyzed while sequencing and again assessed for the next therapy. So we have much more freedom to tailor the treatment for the patients with these new models of NGS. But the problem is NGS is really strong and really fancy technology, but it still faces several challenges which has to be overcome before it can be become a standard tool in the clinical oncology. We have sample quality issues, we have regulatory obstacles, also in consideration with ethical issues regarding storing all the sequencing data. They're still missing restrictive guidelines and SUPs. We have validation problems, also quality benchmarks are missing, as well as which is associated with a quality benchmark. We have minimum requirements for data quality metrics and cutoffs, which still need to be defined. And also the considerations for data storage plays a pivotal role regarding the ethical issues. Who owns the data, for example? And the solution, or the possible solution to overcome these obstacles, is a standardization and a harmonization of workflows, devices, and so on. And this is the second part of my presentation. We can distinguish between crucial parts in NGS which need to be standardized. These can be differentiated between upstream and downstream part. So in the upstream part, we have the sample transport, storage, extraction of the sample, and so on. And this is interesting. So we have to think out of the box, because standardization already starts much before the sample is in the lab. Actually, the sample transport. You know? Does the transport company, do they store the sample properly? Do they have the right temperature? Standards need to become defined for this part. And also after the extraction, after the analysis, then we have the downstream part, so everything which happens after the sample has been sequenced. So the data processing has to be standardized, the annotation, and also the storage of the sequencing results. So those both upstream and downstream pipeline, those are parts of the workflows of an NGS. And then we also can distinguish between tangible and intangible standards. So standard definition for devices or for kits or for reagents, for example, companion diagnostics, which method should be used for this and this tumor entity. There has to become, or there is a need to define standards, and also the intangible standards, which is a bit more complicated. So metrics, quality metrics, minimum thresholds, cutoffs, and parameters. For example, the minimum quality per base call. This is an example. And to just give some suggestions for possible standards and de facto standards, there are already some suggestions present nowadays. So here's again shown the typical sequencing workflows, so beginning with preparing a sequencing library, then preparing and enriching the template, the sequencing, and the final data analysis. And all parts of those workflows, they can be subdivided into substeps. For example, in the step of preparing the sequencing library, we have to control the input at the beginning. So once the sample arrives, we have to take a look what's in that sample, which amount and which quality. And for this, there is the need to determine the quality control checkpoints, for example, using the spectrophotometer or capillary gel electrophoresis, and also standard values or minimum quality metrics for the data generated by those QC checkpoint appliances. For example, you can see down there's a typical electrophoregram. So the result of a capillary gel electrophoresis analysis, and most commonly used is the RIN, the RNA integrity number for RNA samples. And this is a quality value where we can, where we have an idea about the quality of our RNA sample. And standards need to be defined for this value, for example. And also, which is really interesting and plays a very important role, are reference materials. It's actually reference materials for validation of sequencing experiments and sequencing platforms. And here on the right, you can see one possible reference material. This is a genome of the bacteriophage VX174. And actually a reference material in NGS experiments is nothing less than than a very well-known and very well-characterized DNA sample, which is carried along the entire sequencing workflow and undergoes the same handling steps like the original sample. And the idea behind the reference sample is that in the final data analysis, if an error occurred in the reference sample, which is well characterized, then the same error occurred in all likelihood in the main sample. And so then we have to think about which further steps we should take. And so this is also a very important, strong tool for objective comparison between different sequencing platforms. And finally, in the data analysis pipeline, so in the downstream part, there's also the need for the definition of minimum quality metrics for the data analysis. So, for example, the depth of the coverage or the average coverage, especially the depth of the coverage, this is very crucial in clinical application. We need a high coverage, but which coverage is high? What should be a standard value for the depth of the coverage? Also the distribution or the uniformity of the coverage, and the percentage of mapped reads and the average read length, the adapter rate, the mapping quality, also the GC content of the sample, decline in signal intensity, and the minimum quality scores for the base calling. So if you receive our FASTQ files, we have the thread quality score, so per base a quality score, but what should be the minimum requirement for those quality scores? And those questions were already present for some people in the United States who set up standardization efforts. So initiatives have been found, and we're discussing those questions, which I've mentioned before. And so there are already several standardization efforts in NGS present. So let's start just with an overview. The NGS guidelines for the somatic genetic variant detection, which are published by the New York State Department of Health, which contains the same characteristics like published in the Clinical Laboratory Improvement Amendment Guidelines. So those are already quite good known, and a lot of laboratories are certified by CLIA. And those guidelines include validation parameters or performance characteristics, as it is termed by the CLIA. So minimum requirements for accuracy, robustness, precision, repeatability, reproducibility, analytical sensitivity and specificity, as well as reportable and reference range. And furthermore, they also suggested proposals for reference materials for the quality control of sequencing experiments, the same what I've mentioned before. They have made a suggestion for establishment of 3 different controls: the null template control, the negative control, as well as the positive or sensitivity control. Furthermore, there is the National Institute of Standards and Technology, the Genome in a Bottle Consortium. Probably a lot of people also know this consortium. They wanted to investigate reference data, methods, and standards for next generation sequencing. And they proposed the use of well-characterized whole genome sequences as reference materials. And furthermore, they wanted to go more into detail for the use of reference materials or methods for reference materials in order to understand the performance of different NGS systems. Then we have the Association of Biomolecular Resource Facilities, especially the NGS work group. They also worked on similar topics like the identification of optimal methods and strategies for NGS projects, and also the performance evaluation between different NGS platforms. So in order to compare different platforms in their performance. Then one of my favorite work groups, they have published a very comprehensive paper about Accreditation Checklist requirements. This was published by the College of American Pathologists. And this publication includes several detailed standard proposals amongst others for documentation, validation, quality assurance, and so on. So also or a lot of topics which are necessary for accreditation of laboratories, for example. Then furthermore, to come back to the reference materials, there was already one consortium which actively developed RNA spiking controls for NGS experiments. That's the ERCC, the External RNA Control Consortium. So, and then we have the Genomics Standards Consortium, which has published the minimum information about the genome sequence specification. And their aim was to remedy the lack of incomplete genome descriptions for data submission. So now we are deep in the downstream part of NGS experiments. So once all analyses have been accomplished, then the submission of the data to databases is the next step. And there's still the problem that a lot of data gets published without proper descriptions. And therefore, the GSC MIGS, they proposed to you to include in those data also the depth of coverage, the overall quality, the taxonomy, the trophic level, and the propagation as a minimum requirement for the submission of sequencing data. Then we have the microarray quality control project, and especially phase 3 of this project, sequencing quality control. This is really interesting because they also evaluated the technical performance between different NGS platforms by establishing benchmarks with reference samples. So what I've also thought before. Then a quite old initiative, the Human Genome Project, they have published the Bermuda standards, actually. And those are standards for sequence fidelity. And those were one of the first to publish an actual value, an actual quality value as a standard proposal. The Q40, so the quality value in the data analysis, or 99.99% accuracy or 1 error per 10,000 base pairs. They said that should be the minimum quality requirement for sequencing experiments. And then finally, also a very interesting approach and initiative of the next generation sequencing standardization of clinical testing work group. They also published a very comprehensive publication dealing with principles, guidelines, standards, and recommendations for the implementation of NGS into diagnostic laboratories. So actually, exactly this one, which is interesting for precision medicine approaches, especially for standardization of NGS in precision medicine. And furthermore, they have published a novel NGS proficiency testing system, which enables an error identification, which is associated with an FMEA. But what this is, I will go into more detail later on. And the indication of QC problems, so quality control problems. And this PT system also enables the verification of test performance in laboratories. So also a very nice approach. However, so as we have seen, there are a lot of different standardization initiatives already present. So already a lot of work was done, but which was still an obstacle is a lack of documentation. So we have standard proposals, we have proposals for quality metrics, but the documentation of the quality, this is also an issue which is not addressed nowadays. So there's a need to develop procedure operating and inspection and structures, respectively SOPs, and also the establishment of quality records, respectively verification documents for quality proof of data is needed. And those quality records, once developed, are also suitable for providing a quality certificate to the customer, that the customer knows, okay, the sequencing experience was of high quality, so I can rely or I can trust in those results. And one possible approach to establish such a quality documentation is the establishment of a technical note in order to improve the overall quality documentation. And this is actually a piece of paper which is carried along the entire sequencing workflow. And this enables the traceability of error, of errors, and this is also associated with the FMEA, which I will go into detail later on, and the Accreditation Checklist requirements, which were published by the College of American Pathologists. And actually, TN is carried along the entire workflow, as I said, and on each different QC checkpoint, the TN has to be filled by the questions who, what, when, where, why, and how. Like, for example, let's take the fragmentation step in a sequencing workflow. So the processing of the sample, the TN asks for who is fragmenting the sample, so that we have a name, what sample is fragmented. So for example, which order number for which project, when was the sample fragmented? So time and date are also necessary to know. Where was the sample fragmented? So for example, in which laboratory or in which department? Why was the sample fragmented? So this is also, again, order number, so that we can trace the way back to the original sample. And the most important point, how was the sample fragmented? So which device was used or which appliance? So what were the minimum quality requirements for this processing step? And so this represents a certificate for each step, or a quality certificate for the customer, that he knows exactly, okay, what happens with the sample, why and how. And there's a need to establish something like this in NGS experiments. So, and from quality management, if I tell you about quality management, I also have to mention the quality assurance, which is a crucial part of the quality management. And now finally I come to the FMEA, which is actually a quality assurance method. And this is really a really interesting method, actually. The FMEA is just a failure mode and effects analysis. And that's a standard method in preventive quality and risk management. And you can perceive it as a schematic method to recognize and evaluate existing and possible errors, and then finally to determine adequate measures for avoiding these errors. And that means actually you analyze the errors before they happen. And this is important in NGS experience, because if a sample gets failed, you have to start from the beginning. And it also enables the classification of errors. So actually, what is an error in NGS? That's important to define. And I put this picture here of the NASA moon mission in the slide, because this is very interesting. The FMEA originates actually from the NASA. The FMEA was developed prior of the moon mission in order to determine and classify all errors which could occur during the moon mission. Actually, the sense behind this is if one error happens, there's no way. You have only one way to the moon and one way back. So you have to classify and collect all possible errors and define them and define measures or instructions, what happens after the error occurred. And this shows the importance of this tool. And actually, this could be also applied in NGS experiments, but maybe not such as comprehensive, such in the moon mission. And yeah, how does it work? The FMEA requires that for the comprehensive process analysis, and you have to collect all possible error causes in NGS workflows. So what are, for example, some possible error causes? A wrong operation of devices, or also a wear of devices, of old devices, unsuitable analysis processes, or defective reagents or old reagents, unsuitable temperatures during the NGS workflows, and so on. So the list can be continued infinitely. I think there are so many possible error causes in NGS experiments. And so this is the comprehensive process analysis. And after that, after you've identified all errors, you can make these adequate measures or instructions for avoiding these errors. And the idea behind is the FMEA is based on the rule of 10. So that means the cost for troubleshooting performed by correction measures increases tenfold in each stage. So, and that's because the idea of the FMEA to avoid these errors or to classify these errors before they can occur, so that they don't occur. And this is evaluated mathematically by the RPN, that's the risk priority number, and this is calculated by the probability of an error occurrence times the impact or extent of an error times the probability of the error detection. And dependent on the number which will result out there, you have a very detailed classification of an error, and you know in which stages of a workflow you have to be, I would say, very tidy to avoid these errors. So, and that's actually all. Unfortunately, time is not enough to talk about everything what I like to talk, but just to give some more recommendations what is to do, or give some hints and some suggestions for future activities, I wanted to mention some points on my last slide. So the next steps would be also, which are necessary, would be the standardization of the patient evaluation and the electronic medical records, or how they are called nowadays, the electronic health records, especially in consideration of the sequencing data storage. So probably patient's tumor profile gets sequenced. So where do we store those data? Are they in the EMRs or EHRs or databases? So this is a point where we have to think about. Also the EMR or EHR linkage to laboratory results and across health systems is a point which need to be discussed in the future. Then obviously the standardization of clinical practice guidelines when NGS is established into clinical trials. This is an important point. And of course, the consensus finding by frequent expert panels who are specialized on precision medicine is a crucial point. So we have to build a foundation where all stakeholders can discuss about all those issues, find a consensus, and to find finally standards, regulated standards in the end. Then also very important point, biomarker and companion diagnostics validation is important. So therefore, there is need to establish routine interlaboratory tests. So, for example, using sequencing the same NGS regimen with different appliances in different laboratories. So validation concerns are also one point which is necessary in the future. And also the awareness amongst all precision medicine stakeholders. So those awareness has to be raised, it has to be discussed, it has to be marketed, so information has to be shared, that everyone who is associated with the precision medicine knows about the up-to-date information and what is possible and what is not possible. Then a quite simple but nevertheless important point, the terminology. So there's also the need to define official precision medicine terminology definitions. So what is a biomarker? What is a companion diagnostics? What is precision medicine itself? And for example, the ISO, where I was involved in, they have one work group which just deals with definitions, terminology definitions, and biotechnology. So we need official definitions in order to get clarity around this field. And the last point, quite fancy terms which are occurring nowadays quite frequently. We have to keep observe the developments in artificial intelligence and the digital twins. But this is another big chapter, which I will not discuss right now. So I'm at the end. Thank you very much for your attention. And if you have any suggestions, if anything is unclear or you just would like to discuss, feel free to drop me an email or write me on LinkedIn. Thank you very much and take care.
Thank you, Christoph, for that informative presentation. We will now start the live Q&A portion of the webinar. So if you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. The first question is it better to define one general standard or multiple standards for NGS applications in precision medicine?
That's a good question. So it depends on the application, actually. One general standard, only one, would be obviously more advantageous due to better comparability of results and just because of the standardization environments that we have not so much standards. However, one general standard would not be feasible at the moment due to the quite young area and also things like sample and tumor heterogeneity and different requirements between different sequencing platforms. Thus, multiple standards for different sequence regimen will become developed in short to midterm, I would say. And those standards, once established, could become revised or harmonized in long-term developments.
Thank you. The next one is who is or who will be responsible for setting up standards in NGS and precision medicine?
Yeah, this is also a good question. So as I've shown already in the presentation, there are different standardization and quality management initiatives already present. So within the last years, a lot of initiatives have been found. However, the regulated standard structure, this will be determined by official bodies like the International Standardization Organization, the ISO. And what I've mentioned before, there's already one technical committee where I was also involved in within the ISO, and they're advancing standardization efforts in biotechnology. So this technical committee is called ISO TC 276 Biotechnology. And this technical committee comprises already 6 different working groups. And the idea is to merge different experts together to set up standard drafts for, amongst others, biobanks, analytical methods, as well as the data analysis pipelines for NGS. And further, quality organizations like the United States Pharmacopoeia, the USP, they are planning to set up business plans in precision medicine. Also, so for those efforts, voluntary experts are always needed to share their expertise and finding a consensus and finally developing standards. So I can just recommend to get in touch if you're interested in developing those standards. Get in touch with ISO, for example, and your expertise is welcome.
Thank you. Okay, we have time for just one more question. Will NGS advancements turn precision medicine into personalized medicine in the future?
This question corresponds to one of my first slides where I talked about the difference between precision medicine and personalized medicine. So just keep it back in mind. Precision medicine, just better patient stratification, tailored treatments according to environmental, genetic, and lifestyle of different patients. And personalized medicine, one unique treatment for one unique patient. And I think regarding this question, so will NGS turn precision medicine into personalized medicine in the future? Probably yes. But at the moment, the NGS technologies allow just a precision medicine approach, or better said, much better patient stratification down to small populations, which are most likely to benefit from a tailored treatment based on their genetic and genomic profiles. However, the advancements in NGS technologies, also the third generation technologies, so PacBio and Oxford Nanopore, also the biomarker discoveries and the developments in the companion diagnostics, and also the future standardization and harmonization efforts will likely turn precision medicine into personalized medicine, or better said, tailored treatments for each individual. But And in my opinion, I think this will still need quite a long time, because also standardization is a long process, but the future is open.
Well, thank you. I would like to once again thank Christoph Endrullat for his presentation. I'd also like to thank LabRoots for making today's educational webcast possible. Before I go, I want to let everyone know that today's webcast will be available for on-demand viewing through September 21st, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.